Good morning and welcome to Anemia and Chronic Kidney Disease. My name is Molly DeBras and I am the Client Services and Community Manager with the Kidney Foundation of Ohio. A few housekeeping items for today's webinar. Today's webinar will last for 1 hour. Questions can be submitted using the Q&A feature on your screen, either during or directly following today's presentation. And if time permits, those questions will be asked at the conclusion of today's session. Some additional information. For information about future sessions, please visit our website, www.kfohio.org, and save the date. Our 46th Annual Renal Symposium will be happening on September 23rd and 24th. An evaluation will be available following today's session. A little bit about the Kidney Foundation of Ohio. We provide a broad-based program of direct assistance to those with kidney disease, and we promote and provide educational programming for the general public, renal professionals, and patients and their families. We serve a 32-county area in northern and eastern Ohio, and we support 2 affiliate chapters in Lake and Summit counties. We remain the only health and human service agency in Northeast Ohio, which continues to provide direct dollars to patients to assist them through the devastation of their kidney disease. The foundation remains an Ohio-based organization, and all funds raised in Ohio stay in Ohio to help our patients. I'd like to say thank you to today's sponsor, AstraZeneca. And now I would like to introduce Megan Coy. She is a renal sales specialist with AstraZeneca. Megan?
Thank you, Molly. AstraZeneca is really happy to be able to provide this educational presentation today. As Molly said, my name is Megan Coy. I'm the renal sales specialist in the Cleveland East Territory. And I'd like today to introduce Dr. Anil Agarwal, who joins us bright and early from Fresno, California this morning, where he is a nephrologist. Currently, he is the chief of medicine at the Veterans Administration Central California Health System in Fresno, California. He's also professor of internal medicine at the University of California, San Francisco, Fresno. Prior to his role in California, he came from right here in Ohio, in Columbus, Ohio. He was a fellow for the nephrology department in Ohio State, the Ohio State University. He was a chief of nephrology section at University Hospital East. He was also the director of the nephrology interventional program at the Ohio State University, among many other roles. But we like to say you're a Buckeye still, right, Dr. Agarwal? So thank you so much again for joining us. We're really excited to hear about the anemia of CKD, and I'll turn it over to you. Thank you.
Good morning, and thank you very much. Let me go ahead and share my screen. So first of all, I would like to thank you, both Megan and Molly, for welcoming me this morning. And thank you so much to all of you for being here so early in the morning to talk about anemia of CKD, a changing perspective based on the hypoxia inducible factor, or the HIF pathway. So if I can move the slide. A little bit of a technical issue. Okay, now these are my disclosures. I have research support from Akebia and consulting fees from AstraZeneca, Otsuka, and Vascular, and Speaker Bureau of AstraZeneca and Janssen. So first of all, let's examine the scope of the problem. You're all nephrology professionals, so you are familiar with the disease state of anemia of chronic kidney disease. So this is nothing that is a stranger topic to you. So as we all know, anemia is a complication of chronic kidney disease that affects millions of patients in the United States. This is just the United States data. Among patients with chronic kidney disease who have anemia, about 4.8 million that is the population that has anemia with CKD. a low percentage, only less than 23% of patients with anemia of CKD report being treated for it. So anemia is common, but the treatment is not as common. And consequently, at the time of initiation of dialysis, about anywhere from 75% to 85% of patients do have anemia of CKD. So it is exceedingly common state that you are familiar with. Now, the other thing that we need to realize that the anemia burden keeps on increasing with the progression of chronic kidney disease. You can see in this graph that as you go from CKD 3 to 4 to 5, there is a progressive increase in the prevalence of anemia from around 43% in CKD 3, for 64% in CKD 4, and nearly 3/4 in CKD 5. So this is extremely common, keeps on progressing with CKD. And why it is important to realize is that anemia has symptoms. It has a human cost. There is weakness, dizziness, shortness of breath, which all affect the quality of life of the patient who is suffering from that. And not only that, the patients with anemia of CKD who are not on dialysis still have greater healthcare utilization. So 33% more hospital admissions, over 4-fold increase in hematologist visit. This is the data from Medicare population, 20% Medicare sample. So this is essentially people over 65 years of age, this data retrospective data. So exceedingly common condition. Now, the other thing that we need to realize that the severity of anemia is associated with increased morbidity and mortality among patients with CKD. So we know that low hemoglobin level is associated in multiple studies with increased mortality, cardiovascular hospitalizations, and progression to end-stage renal disease. Let us take a look at the data from a study by Thorpe and colleagues that is depicted here in patients with CKD. In this population, there was a comparison made between the non-anemic patients, and they were defined as those with hemoglobin over 12.5, with the anemic patients whose hemoglobin was less than 10.5. And in this study, it was shown that there was a 2-fold increase in the risk of cardiovascular hospitalization, 5-fold increase in risk of death, and 5-fold increase in the progression of kidney disease. So as you can see, as you go from hemoglobin of 12.8 on the left-hand side, you go to 9.4 here, there is a progressive increase in the event rate in these patients. Let's then look at what happens in incident dialysis patients. And as the nephrology professionals all know, incident dialysis is within the first 3 months of starting dialysis. This is a spatially fragile population that is at risk. And this is the data from 2018 United States Renal Data System. And it was shown in this study that the patients at ESRD onset have low hemoglobin and a low erythropoiesis stimulating agent use. So as you can see, in all incident populations of patients on dialysis, the average hemoglobin was 9.3 grams per deciliter. Only about 13% had used ESA. Also, at the time, if you look at the higher risk group, so if you think about this risky population, fragile population, and add more risk to it. So if you pick up the patients with diabetes or hypertension, you will see that still the use of ESA at this point was not as much. And why it is important, when you look at the risk of mortality in this population of incident dialysis patients, the risk of mortality is highest in all dialysis patients within the first year, but especially having 22% mortality within a year. More than a third of these deaths occur within the first 3 months in the incident population. And one of the reasons is that they have a high level of comorbidities. There is cardiovascular disease present in up to 83%, diabetes in 76%, and hypertension in 74%. So not only these patients have a lot of comorbidities, they are already risky. This is a time of transition. This is a very tumultuous point in a patient's life when they start dialysis. And there is no question in my mind that this patient population is very, very much at high risk. And here is a risk factor for mortality that is not being addressed so much. So let's look at then how the treatment of anemia has evolved over last several decades and centuries. So this is very interesting slide. So you might realize that oral iron was used for treatment of anemia as early as 1800s. So anemia is not something that just was recognized in the last 3 or 4 decades. It's been recognized for a long period of time. It may not have been called exactly anemia, but then it was recognized, and oral iron was still used in 1800s. Now, in 1900s, another big advance came almost 100 years later. That was starting the blood transfusions. And in the mid 1900s, the first IV iron was used. That was ferric hydroxide. And it is very interesting to know that all irons now have a shell of carbohydrate to decrease the toxicity of iron, because iron is a very potent oxidizer. So, but in 1900s, they didn't know that, and they used ferric hydroxide, which I'm sure did cause some troubles at the time. And during 1900s, then there was an explosion of other irons. There was high molecular weight dextran, and then low molecular weight dextran, and number of other irons that you are familiar with. Those have started. So there are multiple preparations of iron for oral and intravenous use. Let me focus here on the erythropoiesis stimulating agent. So in 1989, that was a very, very significant year in the life of kidney patients when first time the epoetin alpha was approved for use by FDA in 1989, and that changed the life of dialysis patients. These patients were getting several transfusions every year and had iron overloads and were miserable, living in the hemoglobins of 6, 7, and 8 at the most. And that changed the life of these patients. And short-acting ESA became the standard of care for patients with anemia of CKD. So there now we had 2 ways of treating anemia. That is the iron as well as ESA. So that's what happened. And then there have been some advances since then. In 2000s, there was this long-acting ESA, darbepoetin alpha, then what you know as methoxy, packed epoetin beta, or Mircera that you know now, that came in vogue. And now there are biosimilar ESAs that you are probably using in your dialysis units at this point. So advances have gone, but the paradigm has remained the same. So this is what we used at this time. So ESAs, no question, were a medical innovation. When they were first introduced in 1989, that was a breakthrough treatment in the management of CKD anemia, and they reduced the need for blood transfusion, and consequently also the side effects of blood transfusion, like blood-borne diseases, the infections, hepatitis C was so common, allosensitization for patients who are going to get a transplant, giving them blood can increase the number of antibodies. And of course, every unit of blood comes with iron. So there was also iron overload, and transfusional hemosiderosis was also a risk. So now all the guidelines recommend that you should use ESA to avoid ESA and or iron to avoid transfusion risks. So that was definitely a big advance at the time. But then let's see what happened then. So we all became very adept in using ESAs, IV iron, and we were like, okay, if the patient with hemoglobin of 8 has heart problems and 9 has heart problems, and if they feel better at 10 or 11, or maybe increasing it to 13 or 14 might be even better. But you must have heard that enemy of good is better, and we realize it very hard way. So these are just 3 studies that I'm going to show you today, how if you treat to a higher hemoglobin levels, instead of improving any of the outcomes in these patients with CKD, you can actually make it worse. So these are the 3 studies that are very landmark studies. The first one was the normal hematocrit trial that was done in 1998. These are the patients on dialysis. They had significant heart disease, and over 1,000 patients. Their target hemoglobin was 14. So they were like, these are heart patients. They will do better with higher hemoglobin. So let's compare how they do if their hemoglobin is 14 versus 10. And lo and behold, the outcome was that the death and non-fatal myocardial infarctions increased by 28%. Everybody was in denial. We saw that article in New England Journal of Medicine by Dr. Besarab and his colleagues, and it seemed like, oh no, there is something wrong. This cannot be true. So that led to more trials, and these were a couple of trials that I'm going to show you. One is the Choir study, and another is the Treat study. These were both done in non-dialysis CKD patients, over 1,000 patients in Choir, over 4,000 in Treat. This is the largest study of patients with anemia randomized controlled trial. And especially they said, okay, let's pick up even sicker patients. So let's add diabetes to that. So in Treat study, all the non-dialysis dependent patients with CKD also had diabetes. And the goals were again high, 13.5 in Choir and 13 in Treat, compared to a lower hemoglobin level. And unfortunately, to the surprise of everyone in renal community, these studies also showed that in Choir study, there was a 34% increase in number of deaths, MI, heart failure, and stroke. And the risk of stroke in Treat study was almost double. So that was a shock to the renal community, and that changed how we treated anemia of CKD in everybody. So this not only led to the FDA advising that you should be cautious, and there were black box warnings by the manufacturers. Let me show you a very interesting trend. So this is when the EPO was approved somewhere here in 1989. The top line that you see here in blue is the mean hemoglobin, and then the bottom one, in black, is the top one is the ESA dose, sorry, and the lower one is the hemoglobin level. So because we were all excited about the ESAs, we started giving more ESAs. Look at the dosages that increased from around 10,000 to about almost close to 20,000 units. And then the hemoglobins increased to almost 11.9 grams in 2007. And when those studies that I just described to you were published around 2007, that was around November, when we were all at a ASN meeting, and there were 2 articles back to back in New England Journal of Medicine that shattered everything in what we knew about anemia of CKD, led to black box warnings, and everybody became so cautious about using it. You can see the dosages of ESAs plummeted back to around 9,000, and then the hemoglobins declined from, again, 11.9 to 10.4 by 2016. So that is an interesting historical piece that will help you understand how we have progressed over the years. But fortunately, the science has advanced, and that is a natural cycle. That's what we expect. That's what we want. And at that time, even just the use of EPO and iron in different combinations, sometimes blood transfusion, that became the standard of care in treatment of anemia. But now we knew the risks of that type of paradigm. So in the meantime, the research came that suggested that there is a central regulation of oxygen homeostasis that I'm going to describe to you. And we now know that a complex oxygen sensing cascade, which is known as hypoxia inducible factor, or HIF in short, that pathway drives both the production of EPO and availability of iron for the production of red blood cells. So taking a step back here, just to show you what we learned when we were training. So this is what I learned when I went to med school back almost 40 years ago. So what we knew at the time, as we develop CKD, the kidneys don't produce EPO as much. There are uremic toxins in the blood that decrease the survival of red blood cells. There is also blood loss because of the uremic defects in coagulation. So they may have GI bleeds and other things. They lose blood in the dialysis circuit also. And plus, dialysis is an inflammatory state, and there is iron deficiency because patients don't eat. That was essentially what we knew at the time. So low iron, low ESA, and low half-life of red cells, that was it. But now we know that there is also increased hepcidin that leads to further iron restriction or iron deficiency, and there is impaired oxygen sensing activity in the kidneys, which may sound a little surprising, but basically that tells you that there are multiple pathophysiologies of anemia of CKD. So let's talk about inflammation. By nature, CKD is a state of inflammation. As there is an accumulation of inflammatory factors, and what that does is that there is suppressed endogenous EPO production. So when you look here at the top, inflammation limits the erythroid proliferation and maturation, and the kidneys do not secrete as much EPO. There is decreased red cell span, as I told you. At the same time, there is also increase in production of hepcidin by the liver in response to inflammation, and also because the kidneys are not able to metabolize or excrete hepcidin, so there is a higher level. And I will talk about hepcidin in a minute and how that ties down to how your iron is utilized and is available. So all these all lead to low plasma iron, low EPO, and decreased erythropoiesis that makes anybody anemic. So now let's talk about hepcidin a little bit. So hepcidin is a very small peptide that is produced in the liver that regulates iron availability in the body. Just to give you just a little piece of history, it was discovered right around 2000, and it was known as the liver expressed antimicrobial peptide. It was thought to be a part of the immunity to protect against infections from outside, because some bacteria and fungi utilize iron, and if you restrict the iron, they will not grow so much. So there was secretion of hepcidin. So in CKD, as I told you, hepcidin is elevated because there is more production and there is decreased renal clearance. What hepcidin does is it inhibits the ferroportin. It down regulates ferroportin. What is ferroportin? This is the only way. This is like a channel from the cell where the iron might be inside the cell. The only way the iron can get outside the cell is through this channel of ferroportin, unless you destroy the cell and the iron will come out. But in a living cell, the only way it comes out is by ferroportin. So normally it can come out, but when you have hepcidin, it will not, it will degrade the ferroportin or downregulate it. So there will be no channel for iron to come out, and there will be iron sequestration. At the same time, because this is the only mechanism to transport iron, it also decreases absorption from the gut, there's more secretion of hepcidin that leads to sequestration of iron within the cells, within the macrophages, and within the liver and spleen, reticuloendothelial system. And even though you may have good stores of iron inside the body, you will end up not having available iron to create red cells. So next, a new thing that we now know, that there is impaired oxygen sensing in CKD that reduces EPO production. We will talk in more detail as we go on, but basically, if you don't sense hypoxia in the kidney, you are not going to produce that much EPO, and at the same time, you have poor iron availability in these patients. So combination of EPO deficiency and iron deficiency or iron restriction that leads to development of anemia of CKD. So let me show you a very nice video here.
Anemia, the oxygen sensing mechanism in the kidney responsible for EPO production, is dysregulated. This mechanism resides in the renal erythropoietin producing cells and involves a hypoxia inducible factor, or HIF pathway, and prolyl hydroxylase enzymes. In healthy individuals, in the presence of sufficient oxygen, prolyl hydroxylase enzymes rapidly hydroxylate HIF-alpha, tagging it for proteasomal degradation. Under hypoxic conditions with insufficient oxygen available, hydroxylation and degradation of HIF-alpha do not occur. Thus, HIF-alpha levels increase, and HIF-alpha enters into the nucleus to bind with HIF-beta. This activates the HIF pathway and leads to target gene transcription. In CKD anemia, the oxygen
I'm trying to get my slide moving here. All right, so now you saw that video, and now you know how the hypoxia inducible factor works. So what is hypoxia inducible factor? So as you can see, these HIFs are oxygen sensitive proteins that regulate the transcription response of cells to hypoxia. So hypoxia inducible factor, there's this very interesting history that you will notice. This was first identified by Semenza and Wong in the early 1990s as a DNA transcription factor that is induced by low oxygen levels that initiate EPO synthesis. Since then, the role of HIF has been well characterized by a number of different groups. The HIF pathway is essentially a master regulator. It's the central regulator of the cellular response to hypoxia, which coordinates a physiological response leading to erythropoiesis by transcribing the target genes. And transcription of these genes produces proteins that maintain erythropoiesis. So in essence, what it does, it increases EPO production. It also, so it's a very, very nice symphony here. So what it does, it increases EPO production, but it will not be useful if there was increased hepcidin. So it also suppresses hepcidin. It will not be useful if there was no iron. So it increases enteric iron absorption and transport, and also allows redistribution of iron stores from inside the cells to outside so that you can utilize for erythropoiesis. So in a way, everything fits together very well once you have the HIFs. So now also let's look at how the studying EPO regulation led to the discovery of HIF pathway. So this is a very interesting history. In 1800s, people realized that when you have low oxygen, there is an increased red blood cell production. They noted that people who lived at altitudes had higher hemoglobin levels, and towards the late 20th century, in 1977, finally they figured out that there is a human EPO that they were able to purify. Remember, it was approved in 1989. In 1985, they were able to clone the EPO gene. 1993, here there was something very significant that happened. Dr. Semenza and Dr. Wong identified HIF as the oxygen sensing regulator of the EPO. And in 2001, there was a group led by Dr. Bill Callan and Radcliffe that elucidated the mechanism of HIF degradation by prolyl hydroxylase enzyme. So there is an enzyme that degrades the HIF in normoxic conditions. And in 2019, Semenza, Callan, and Radcliffe, just 2 years ago, Semenza, Callan, and Radcliffe, two of these were nephrologists. They were named as they received the Nobel Prize in medicine for discovery of this pathway that may lead to some newer therapeutic possibilities. So let's take a final look at the oxygen regulation and the kidneys in people with healthy kidneys. So let's talk about the healthy kidneys first before we start looking at the diseased kidney. So the kidneys are the key organs, and this might surprise you. Kidneys are the key organs, not the heart, not the lung, that sense oxygen in the body that leads to increased erythropoiesis. So although the oxygen demand to maintain the kidney tissue is modest, but we know that there is ischemia inside the normal kidney also. There is a lot of work that these kidneys do. So you filter a lot of fluid. You know that there is GFR of roughly about 120 cc's per minute. That leads to a lot of filtration that needs to be reabsorbed. And the sodium that is filtered with it very freely has to be reabsorbed also, because you need sodium to maintain your volume. So that leads to a lot of utilization of iron within the kidneys. This is a very oxygen intensive process that requires a lot of oxygen. So this high oxygen demand is matched by the oxygen supply normally. So there will be enough oxygen and you don't need the EPO if that is the case. But if there is even slight decrease in the oxygen supply to the kidney to be able to do this sodium reabsorption, that will result in hypoxia. Now, when there is hypoxia, it is sensed by a special group of cells known as the REP cells, or the renal erythropoietin producing cells. Now, remember, I did not know anything about these for about last 5-6 years. This is all new research that has come up. So now we recognize that there are REP cells that actually sense hypoxia within the kidneys. So then let's take a look at the HIF signaling. So how does the hypoxia inducible factor works with the REP cells? So the HIF pathway is really the oxygen sensor of the kidneys. In normal conditions with sufficient diffused oxygen, there are prolyl hydroxylase enzymes. These enzymes hydroxylate the HIF alpha, as you saw in the video, at 2 sites. Now, what that does, it tags the HIF molecule to the von Hippel-Lindau protein, known as VHL protein. And that is then picked up by ubiquitin. And you may know that ubiquitin is something that is present almost everywhere, which takes these items that are tagged to the proteasome. Proteasome is really the trash can in the cell, so it takes it there and destroys it. So that's the end of it. If you have good oxygen, you don't need EPO, you don't need HIF, and it gets taken care of. Done. No HIF signaling that takes place. But if you have hypoxia, say there is lack of oxygen getting there for whatever reason, the hydroxylation, those enzymes, they do not work. And when they do not work, that gives the opportunity, as you saw in the video, I'm reminding you, the HIF alpha dimerizes with the HIF beta, combines with it, and then activates the target of gene expression within the cell, and that leads to EPO production that restores the blood oxygen level. So I hope it makes sense. If you have good amount of oxygen, you don't need HIF. If you have less, that's of course you're going to need it. So that enzyme doesn't work, and then you can use that to dimerize and produce more EPO. Now, what else does HIF pathway do? So hypoxia is a life-threatening event. So it cannot be just doing one thing. It has to do multiple other things. So the HIF pathway that regulates EPO production in the kidney also first regulates the iron availability, because it wouldn't make sense to increase HIF and not have iron. It allows for a coordinated supply of factors that are required for erythropoiesis. So you will have iron, you will have EPO, and that will lead to better erythropoiesis, and there will be improved iron absorption, mobilization, and transport. So it kind of takes care of the whole entire erythropoiesis process. So what happens when the kidney function is impaired? So in CKD, this change in oxygen consumption disrupts HIF signaling. So how does the balance get off in CKD? So as you know, as the GFR declines, you cannot absorb as much sodium. When you cannot do that much sodium transport, you don't need that much oxygen because you're not using that much oxygen. Now that leads to reduction in the HIF signaling. So you don't get that much HIF signaling. And consequently, if you don't have HIF signaling, the rep cells, the renal erythropoietin producing cells, feel that there is a pseudo-normoxia. So it's not really normal, but then the kidney says, okay, we have enough oxygen, even though it is low, but it meets the kidney's needs to absorb sodium. And then the REP cells are not stimulated. And when they're not stimulated, you get decreased EPO production, and there is insufficient erythropoiesis. You cannot correct hemoglobin levels. There is worsening of anemia. There is decreased oxygen delivery to the body. So hope that makes sense. That when the kidneys are not really working as much, you really don't need that much HIF or EPO, and that worsens the anemia even further. So let's take a look what then happens when there is reduction of oxygenation of critical organs. So you have red cell production, there is anemia. Delivery of oxygen to the rest of body is reduced, and this may lead to end organ damage. So essentially, over a period of time, you may get left ventricular hypertrophy, there is poor cognition, the brain activity is reduced. So all that happens with that. So now you realize that what we were taught in med school a long time ago, that it's just EPO deficiency and iron deficiency. That's certainly not true. So we have multiple other things going on. There is impaired oxygen sensing, there is presence of inflammation, there is elevated hepcidin that locks up the iron inside the cell, it cannot get out. And the problem with that paradigm is that the problem is we only have currently what we know how to treat anemia is give EPO, because it replaces that hormone that really works to increase erythropoiesis. And also we only know to give IV iron, but we know there is inflammation, there is hepcidin, and there is decreased oxygen sensing in the kidneys. Does any of these treatments that we have right now address those problems? I don't think so. So we can replace EPO, yeah, we can replace iron, but we are not doing anything to the oxygen sensing or inflammation or elevated hepcidin. So current guidelines recommend that the treatments serve to supplement inadequate levels of EPO and iron in patients with anemia of CKD. But these therapies offer a very constrained clinical effect. without addressing the factors of inflammation and high hepcidin level or the oxygen sensing. So we do not have a very effective treatment, but I definitely wanted to let you know of our understanding of the new things that we are learning these days. And that is definitely very helpful in hopefully formulating something in future. Now, to summarize my talk, I hope I have made it clear to you that anemia of CKD is very common, and it is a significant burden, both in sense of what resources we have, what is the human cost of it, and what is financial cost of it. So that's a problem that is still an unmet need, in other words, and we need to have something hopefully in future that will address some of those issues. Supplementation with ESA and iron, yes, it works. It's not like it doesn't work. We were able to raise hemoglobins by those two treatments. And when we gave tons of EPO, tons of iron, we were able to increase the hemoglobin levels even higher. But that does not mitigate the effects of inflammation and high hepcidin levels. In healthy individuals, activation of HIF pathway can do something naturally without giving external erythropoietin that initiates a cascade of events that stimulate coordinated erythropoiesis. Coordinated because there is EPO, there is more iron absorption, it addresses hepcidin, addresses inflammation to some extent, and then improves iron utilization and reduces hepcidin levels. So that is something that is very important. And now we also know that activation of HIF pathway is disrupted in patients with anemia of CKD because in these patients, there is poor oxygen sensing there because of the pseudonormoxia that I told you. And so it is now very, very clear to us that anemia of CKD is more than what we knew, unfortunately, for a very long time. It's not just EPO deficiency and iron deficiency. So I just wanted to go over this disease state and wanted to tell you what are the new things in the disease state of anemia of CKD. And I will be happy to take any question. Thank you so much for your time. And please feel free to ask any questions. Thank you. Thanks again. Back to Megan and Molly.
Okay, thank you so much, Dr. Agarwal. That was a wonderful, very informative presentation. I personally learned a lot, especially that was about the best overview I've ever gotten of how oxygen, really plays a role in anemia and in the kidneys. And I will put it out to the audience. If you have any questions, please put them in the chat or the Q&A, and I will announce them to the doctor. I do have one question, and now having learned all of this information, it kind of presents a very clear explanation as to how COVID, is particularly impacting kidney patients because of, with COVID, impacting oxygen levels in patients, this in turn then can cause kidney failure. Is that an accurate assessment based on the HIF pathways?
So there are a number of pathophysiologies of, that's an excellent question, by the way. I mean, it's very timely, and unfortunately the world is suffering from COVID. We have gone through this, and European countries, and now India is having the worst pandemic there. There are multiple pathophysiologies of kidney failure in COVID, not just the oxygen sensing issue. There is direct damage to the podocytes in the kidneys. And of course, the patients are septic and they're getting a lot of toxic medicines. The number of etiologies, and I'm sure hypoxia sensing mechanism also plays a role in that.
Great, thank you for that. We'll give it another minute or two. If anyone else in the audience has a question, again, please submit them in the Q&A and the chat features. We'll just give it a minute. Actually, if you don't mind, Doctor, I have another question.
Sure.
So you had mentioned REP cells. Are those just specific to the kidneys, or are those type of cells, and again, forgive me, I'm not a doctor, are those type of cells like found in the other organs, like the liver as well too, with, because you know, with your presentation, it was very how interconnected all of the different organs are. So are REP cells like just limited to the kidneys, or are those type of cells found in other organs as well too?
Well, that's an excellent question again. So very, very relevant. Yeah, so you might wonder if you stimulate the HIF pathway in somebody who has kidney failure. I think that's what you're pointing out, is that if you have a patient with complete kidney failure, the kidneys are not working, would you still be able to stimulate the RAP cells somewhere else maybe and produce EPO? So it has been shown that even in patients who do not have any kidneys, the kidneys have been taken out, there can be increase in the EPO production. And in those people, once the kidneys are gone, so first of all, even the diseased kidney in dialysis patient may have some cells left that might still be able to produce. But even if there are no kidneys and zero kidney function, no RAP cells there, the major source of erythropoietin in those patients comes from liver. So it must be the liver that has the RAP cells. There are many other organs that have their own system and may have RAP cells. So there may be some in heart or brain as well, and the vascular endothelium. The main one that is responsible for majority of the EPO production, that comes from liver. And thank you, Dr. Zakarian. It's glad to have you in the audience. Miss you too. Thanks. So there is another question there in the chat. And so in a newly diagnosed person with renal failure grade 4, so it's, I think you're referring to the CKD stage 4, GFR between 15 and 30, related to vasculitis, hemoglobin around 8.5 and steady there for 6 weeks. When is traditional treatment recommended? Well, so this is a great question. So you have a patient who has CKD4, hemoglobin is low, and it's consistently low. It's not like one reading. I think it's a very reasonable time when you can initiate treatment for anemia. Now, let me just tell you that generally you do not, the current guidelines are that you do not start treatment unless it is below 10. So it's definitely below 10, or the patient has symptoms. Even below 10, you don't have to start ESA if there are no symptoms. So I would say 8.5 is a decently low level where there is this patient may be at risk of transfusions. If something happens, say the patient is on a steroid treatment for vasculitis, develops an ulcer and has GI bleed, of course, I mean, this patient can require a red cell transfusion in a minute. So this patient is at a high risk of transfusion. I would recommend that you start looking at the iron and start ESA as well. So I think you have to connect these clinically, but this sounds like a good place to consider starting treatment with an ESA. Is there another question there?
I am not seeing any follow-up questions. If anyone does have a question, again, please put it in the chat or the Q&A. But I think we'll just wrap it up for right now. And if you do have any other questions, please, let us know. We can forward them to the doctor. Thank you again for your participation. It was a fabulous webinar. And I think we'll just wrap it up. So thank you again, Doctor and Megan. Have a great day, everyone.
Thank you so much, everyone, for attending. Thank you for inviting. Thanks, Megan.